living matter of the cell, that is protoplasm, contains a number of organic and inorganic compounds. A number of metabolic reactions are carried out in this living mass, that is protoplasm. These metabolic reactions are very important for carrying out different functions in the cell. Students, you know, all these reactions are controlled by special type of proteins called enzymes. These enzymes have an extraordinary catalytic power, and today we are going to study them in detail.
ਵਿਦਿਆਰਥੀਓ, ਤੁਹਾਡਾ ਅੱਜ ਦੀ ਕਲਾਸ ਵਿੱਚ ਸਵਾਗਤ ਹੈ। ਸਾਡਾ ਅੱਜ ਦਾ ਚਰਚਾ ਦਾ ਵਿਸ਼ਾ ਹੈ enzymes, properties, classification and mode of enzyme action. Enzymes are water soluble, large proteinaceous macromolecules. Enzymes help to catalyze metabolic reactions. Although W. Kuhan described about existence of enzymes for the first time, but Buckner extracted enzymes for the first time from yeast cells. शब्द enzyme दो ग्रीक शब्दा en पाव in ਅਤੇ zyme पाव yeast तो लिया गया है. So meaning of word enzyme is equal to inside the yeast. Enzymes are studied under the branch enzymology. These enzymes are very important as they are one of the keys to understand how cells survive and proliferate. Enzymes catalyze hundreds of stepwise metabolic reactions by which nutrient molecules are degraded, chemical energy is conserved and transformed. Enzyme-controlled reactions are basic to all phenomena of life, that is respiration, digestion, growth, muscle contraction, nerve impulse conduction, etc. ਅੱਜ ਦੀ ਕਲਾਸ ਵਿੱਚ ਅਸੀਂ enzymes ਦੀ nature, properties and enzyme ਦੇ action ਬਾਰੇ ਗੱਲ ਕਰਾਂਗੇ। ਪਰ ਵਿਸਥਾਰ ਵਿੱਚ ਜਾਣ ਤੋਂ ਪਹਿਲਾਂ ਮੈਂ ਤੁਹਾਡੇ ਨਾਲ ਅੱਜ ਦੀ ਕਲਾਸ ਦੇ ਲਰਨਿੰਗ ਆਬਜੈਕਟਿਵਸ ਸਾਂਝੇ ਕਰ ਲਵਾਂ।
On completion of this topic, learners will be able to describe the nature of enzymes, enlist the properties of enzymes, give the classification of enzymes, describe the mechanism of enzyme action, differentiate enzymes and inorganic catalysts, discuss various factors affecting enzymes, describe importance of enzymes.
ਵਿਦਿਆਰਥੀਓ, enzymes ਖਾਸ ਤਰ੍ਹਾਂ ਦੇ ਪ੍ਰੋਟੀਨਸ ਹੁੰਦੇ ਹਨ ਜੋ ਕਿ ਸੈੱਲ ਦੇ ਵਿੱਚ ਕੈਟਾਲਿਟਿਕ ਐਕਸ਼ਨ ਪਰਫਾਰਮ ਕਰਦੇ ਹਨ। ਇਹਨਾਂ ਦਾ origin ਬਾਇਓਲੋਜੀਕਲ ਹੁੰਦਾ ਹੈ। ਇਸ ਕਰਕੇ enzymes metabolic reactions which biocatalyst ਵਜੋਂ ਕੰਮ ਕਰਦੇ ਹਨ। So enzymes are proteinaceous substances which catalyze biochemical reactions, but they remain unchanged during the reaction. That means they are not consumed in the reaction. So enzymes are proteinaceous substances which catalyze biochemical reactions and remain unchanged towards the end of reaction. ਸੈੱਲਾਂ ਵਿੱਚ ਕੁਝ substances ਹੁੰਦੇ ਹਨ ਜੋ ਕਿ catalyst ਵਜੋਂ ਕੰਮ ਕਰਦੇ ਹਨ। ਇਹਨਾਂ biocatalyst ਨੂੰ enzymes ਕਿਹਾ ਜਾਂਦਾ ਹੈ। These enzymes are synthesized inside the cells. ਸੈੱਲ ਵਿਚਲਾ DNA enzymes ਦੀ production ਨੂੰ control ਕਰਦਾ ਹੈ। Some enzymes are used in a cell where they are produced and are called as endoenzymes. But sometimes enzymes come out of cell and act somewhere else and are called as exoenzymes. Chemically, all enzymes are proteins, but all the proteins are not enzymes. With the exception of a small group of catalytic RNA molecules, all enzymes are proteins. So let us now understand the nature of these enzymes.
Chemical nature of enzymes. Most enzymes are proteins. Some enzymes may contain additionally a non-protein group. So enzymes can be simple or conjugated. Simple enzymes are made of proteins only. Example, pepsin, trypsin, urease, etc. Conjugated enzymes consist of two parts, a protein part called apoenzyme and a non-protein part called cofactor. Cofactors can be organic or inorganic in nature. Organic factors are of two types: prosthetic groups, which are tightly bound to apoenzyme. Example, heme is the prosthetic group of catalase enzyme. Cofactors are associated with apoenzyme only during the process of catalysis. Example, FAD and NAD. Inorganic cofactors are metal ions. Example, magnesium ions for hexokinase, zinc ions for carbonic anhydrase.
ਭਾਵੇਂ enzyme molecule ਦਾ ਸਾਈਜ਼ ਵੱਡਾ ਹੁੰਦਾ ਹੈ, ਪਰ ਇਸ ਦਾ ਥੋੜਾ ਜਿਹਾ ਹਿੱਸਾ ਹੀ ਐਕਟਿਵ ਹੁੰਦਾ ਹੈ। ਇਸ ਐਕਟਿਵ ਹਿੱਸੇ ਨੂੰ ਐਕਟਿਵ ਸਾਈਟ ਕਹਿੰਦੇ ਹਨ। ਇਸ ਕਰਕੇ enzyme ਦੀ ਐਕਟਿਵ ਸਾਈਟ ਉਹ ਸਾਈਟ ਹੈ ਜੋ ਕਿ ਇਸ ਦੀ ਸਪੈਸੀਫਿਕ ਸ਼ੇਪ ਅਤੇ ਸਾਈਜ਼ ਦੇ ਕਾਰਨ ਪਾਰਟਿਕੂਲਰ ਸਬਸਟ੍ਰੇਟ ਮੋਲੀਕਿਊਲ ਨੂੰ ਅਟਰੈਕਟ ਅਤੇ ਹੋਲਡ ਕਰਨ ਦੇ ਸਮਰਥ ਹੁੰਦੀ ਹੈ। ਇਸ ਸਾਈਟ ਵਿੱਚ ਕੋਈ ਹੋਰ ਮੋਲੀਕਿਊਲ ਦਾਖਲ ਨਹੀਂ ਹੋ ਸਕਦਾ। The substrate that fits into its binding site is called ligand. The inactive precursor of enzyme is called proenzyme. Enzymes ਦੀ chemical nature ਬਾਰੇ ਪੜ੍ਹਨ ਤੋਂ ਬਾਅਦ ਆਓ ਹੁਣ enzymes ਦੀਆਂ properties ਜਾਂ characteristics ਬਾਰੇ ਵਿਚਾਰ ਕਰੀਏ।
Properties of enzymes. Protein nature. Enzymes are polymeric globular proteins. They have also additional non-protein substance associated with their activity. Molecular weight. Enzymes are giant molecules with high molecular weight. They have molecular weight ranging between 6,000 to 46 lakhs. Chemical activity. Enzymes do not initiate the reaction but accelerate the reaction. Unchanged form. Enzymes combine temporarily with substrate molecule but are not consumed or changed permanently in the reaction. Reversibility. Enzyme-controlled reactions are reversible reactions. Enzymes affect only the rate of biochemical reactions, not the direction. Efficiency. Most enzymes have a high turnover number, which is the number of substrate molecules changed per minute by an enzyme. The higher the turnover number, the more efficient the enzyme is. For example, a molecule of catalase from cattle liver decomposes 50 lakh molecules of hydrogen peroxide to water and hydrogen in 1 minute at 0 degrees Celsius. It means turnover number of catalase is 50 lakhs at 0 degrees Celsius. Temperature sensitivity. The enzymes are thermolabile, that is, heat sensitive. They function best at an optimum temperature. Substrate specific. Enzymes are specific in action. For example, maltase, sucrase, and lactase can hydrolyze maltose, sucrose, and lactose. pH sensitivity. The enzymes show maximum activity at an optimum pH, that is 6 to 8. Their activity slows down with decrease and increase in pH.
Well, students, now you must have become familiar with enzymes, which are characterized by giant molecules having high molecular weight, proteinaceous in nature, have high turnover number, and are substrate specific. So now we will discuss major aspect of enzymes, that is how they are classified. According to International Union of Biochemistry or IUB, enzymes may be oxidoreductases which take part in oxidation and reduction reactions, transferases which bring about transfer of a group from one molecule to another, hydrolases cause addition of water molecule to a variety of bonds resulting in the cleavage of the substrate. Lyases catalyzes the cleavage of specific covalent bonds. Isomerases catalyzes the rearrangement of molecular structure to form isomers. Ligases. This enzyme catalyzes the joining of two substrates to form a large molecule. ਵਿਦਿਆਰਥੀਓ, ਤੁਸੀਂ ਨੇਚਰ, characteristics ਅਤੇ classification ਬਾਰੇ ਪੜ੍ਹਿਆ ਹੈ. Now you must be curious about the working of enzymes, that is, how they act to catalyze the reaction. Now we will discuss about mechanism of enzyme action.
Mechanism of enzyme action. Lock and key hypothesis. This hypothesis was put forward by Emil Fischer in 1894. You know, students, how lock and key works. Just have a look. This method is based on this lock and key principle only. Each enzyme has a specific site known as active site upon its surface. The surface configuration of enzyme is such that only a substrate with matching configuration can fit into this active site. It is just similar to putting a key into a lock. When substrate binds to active site, ions get transferred to substrate. It forms an unstable enzyme-substrate complex. Soon, this enzyme-substrate complex breaks to release a product, as you can see here in the visual.
ਵਿਦਿਆਰਥੀਓ, ਹੁਣ ਤੁਹਾਨੂੰ ਪਤਾ ਲੱਗ ਹੀ ਗਿਆ ਹੈ ਕਿ ਪ੍ਰੋਡਕਟਸ ਬਣਾਉਣ ਲਈ ਸਬਸਟ੍ਰੇਟ ਮੋਲੀਕਿਊਲ ਤੇ ਐਂਜ਼ਾਈਮ ਕਿਸ ਤਰ੍ਹਾਂ ਕੰਮ ਕਰਦੇ ਹਨ. ਐਂਜ਼ਾਈਮਸ ਦੇ ਮੋਡ ਆਫ ਐਕਸ਼ਨ ਨੂੰ ਐਕਸਪਲੇਨ ਕਰਨ ਲਈ ਇੱਕ ਹੋਰ ਹਾਈਪੋਥੇਸਿਸ ਹੈ.
Induced fit hypothesis. It was proposed by Koshland in 1959. You know, students, how our hand fits into a glove. This method can be compared to a hand slipping into a glove, which then causes or induces a fit. The active site of enzyme has two groups, buttressing group and catalytic group. Buttressing group is meant for supporting the substrate, whereas the catalytic group is able to weaken the bonds of molecules by electrophilic and nucleophilic forces. When the substrate molecules bind to the enzyme molecule, a change is brought about in the active site to precisely fit the transition state. This induced fit holds the substrates at the correct angle for the reaction to take place, as you can see here in the visual. This forms an enzyme-substrate complex. The substrate is converted into products as shown here.
Students, now it must be clear to you how an enzyme-substrate complex is formed and how products are produced and enzyme come out unchanged at the end of reaction. ਤੁਸੀਂ ਜਾਣਦੇ ਹੋ ਕਿ enzyme ਦੀ activity ਕਈ factors ਨਾਲ ਪ੍ਰਭਾਵਿਤ ਹੁੰਦੀ ਹੈ. These factors are temperature, pH, poisons, substrate concentration, product concentration, and inhibitors. Let us discuss them one by one.
Temperature. Rate of reaction increases with increase of temperature. Rate of enzyme catalyzed reaction is minimum at low temperature. Above the optimum temperature, the activity of enzyme decreases as it becomes denatured. pH. Some enzymes are active in acidic pH, some in alkaline pH, and some in neutral pH. Each enzyme has optimum pH at which it acts best. Substrate concentration. Increase in substrate concentration increases the rate of chemical reaction. This increase in rate is due to occupation of more and more active sites by the substrate molecule. Higher number of collisions take place between the substrate molecules. Product concentration. Accumulation of end products lowers the enzyme activity. So the products are quickly removed from the site of formation and reaction does not suffer. Poisons. Denature the enzymes and make them ineffective. Inhibitors. Enzymes are denatured by certain agents that inhibit the action of enzymes. Such agents which inactivate the enzymes are called inhibitors. Iron, copper, and mercury ions are common inhibitors.
Other than these factors, there are some activators such as some metallic inorganic ions which also influence the activity of enzymes and act as pro-enzyme activators. विद्यार्थियों, mode of enzyme action, characters of enzymes ਅਤੇ factors affecting the enzyme action ਬਾਰੇ ਪੜ੍ਹਨ ਤੋਂ ਬਾਅਦ ਆਓ ਵੇਖੀਏ ਕਿ enzymes ਸਾਡੇ ਰੋਜ਼ਾਨਾ ਜੀਵਨ ਵਿੱਚ ਕਿਵੇਂ ਮਹੱਤਵਪੂਰਨ ਹਨ. Enzyme control reactions are basic to all phenomena of life like respiration, growth, muscle contraction, photosynthesis, digestion, etc. Enzymes are commercially used for cheese making, detergent manufacturing, food processing, waste treatment, etc. Enzymes play an important role in food preparation, cleaning, etc. Enzymes are used in fermentation process to make alcoholic beverages. Students, though enzymes catalyze a number of reactions and act as catalyst, but they basically differ from inorganic catalyst. So let us now check out the differences between enzymes and inorganic catalyst.
Differences between enzymes and inorganic catalyst. Inorganic catalysts are inorganic in nature, whereas enzymes are proteinaceous in nature. Catalysts are small molecules, whereas enzymes are complex macromolecules. Catalysts are less efficient, whereas enzymes are highly efficient. Catalysts are not sensitive to small changes in temperature, whereas enzymes are effective in certain range of temperature. Catalysts have low molecular weight, whereas enzymes have high molecular weight. Poisons do not have any adverse effect on catalyst, whereas enzymes are affected adversely by large number of protein poisons.
Enzymes ਅਤੇ catalysts ਕੁਝ ਹੱਦ ਤੱਕ ਇੱਕ ਦੂਜੇ ਨਾਲ resemble ਕਰਦੇ ਹਨ. Both remain unchanged at the end of reaction. Both are required in very small quantities. They don't alter the equilibrium of reaction. They do not initiate a reaction but only enhance the rate of reaction by lowering down the activation energy. ਮੈਨੂੰ ਉਮੀਦ ਹੈ ਕਿ ਤੁਸੀਂ enzymes, properties, mode of action ਅਤੇ factors affecting enzymes ਬਾਰੇ ਕਾਫੀ ਕੁਝ ਜਾਣ ਗਏ ਹੋ. Let us summarize the entire topic. Enzymes are water-soluble, large proteinaceous macromolecules. Enzymes can be simple or conjugated. Simple enzymes are wholly made of proteins. For example, pepsin, trypsin, urease, etc. Conjugated enzymes consist of two parts, a protein part called apoenzyme and a non-protein part called cofactor. Enzymes are polymeric globular proteins. Enzymes are giant molecules with high molecular weight. Enzymes do not initiate the reaction but accelerate the reaction. Enzymes are not consumed or changed permanently in the reaction but come out unchanged at the end of reaction. The enzymes are thermolabile, that is, heat sensitive. Enzymes are specific in action. The enzymes show maximum activity at an optimum pH, that is 6 to 8. Enzymes can be classified as oxireductases, transferases, hydrolases, liases, isomerases and ligases. The two hypotheses for mode of enzyme action are lock and key hypothesis, induced fit hypothesis. Lock and key hypothesis was put forward by Emil Fischer in 1894. Induced fit hypothesis was proposed by Koshland in 1959. The active site of enzyme has two groups, buttressing group and catalytic group. Buttressing group is meant for supporting the substrate, whereas the catalytic group is able to weaken the bonds of molecules by electrophilic and nucleophilic forces. activity of enzymes is affected by a number of factors like temperature, pH, substrate concentration, product concentration, inhibitors, and poisons. Though enzymes catalyze a number of reactions and act as catalyst, but they basically differ from inorganic catalyst. ਹੁਣ ਅਸੀਂ ਇਹ ਪਤਾ ਕਰਦੇ ਹਾਂ ਕਿ ਤੁਸੀਂ ਅੱਜ ਦੀ ਕਲਾਸ ਵਿੱਚ ਕਿੰਨਾ ਧਿਆਨ ਦਿੱਤਾ ਹੈ। ਮੈਂ ਤੁਹਾਡੇ ਤੋਂ ਕੁਝ ਪ੍ਰਸ਼ਨ ਪੁੱਛਾਂਗੀ, ਪਰ ਮੇਰੇ ਉੱਤਰ ਦੇਣ ਤੋਂ ਪਹਿਲਾਂ ਤੁਸੀਂ ਵੀ ਮੇਰੇ ਵੱਲੋਂ ਪੁੱਛੇ ਗਏ ਸਵਾਲਾਂ ਦਾ ਜਵਾਬ ਦੇਣ ਦੀ ਕੋਸ਼ਿਸ਼ ਕਰਨਾ। ਮੇਰਾ ਪਹਿਲਾ ਸਵਾਲ ਹੈ, What are enzymes? The answer is, enzymes are proteinaceous substances which catalyze biochemical reactions and remain unchanged towards the end of reaction. My next question is, what are conjugated enzymes? Answer. Conjugated enzymes consist of two parts, a protein part called apoenzyme and a non-protein part called cofactor. Next question. Name the classes into which enzymes have been grouped. Answer. Enzymes can be classified as oxireductases, transferases, hydrolases, liases, isomerases, and ligases. My next question is, define endoenzymes and exoenzymes. The answer is, some enzymes are used in a cell where they are produced and are called as endoenzymes. But some enzymes come out of cell and act somewhere else and are called as exoenzymes. The next question is, name two hypotheses for mode of enzyme action. Answer. The two hypotheses for mode of enzyme action are lock and key hypothesis and induced fit hypothesis. Next question. Who gave lock and key hypothesis? Answer. This hypothesis was put forward by Emil Fischer in 1894. Next question. Who proposed induced fit hypothesis? Answer. It was proposed by Koshland in 1959. The next question is, list two active sites of enzyme. Answer. The active site of enzyme has two groups, buttressing group and catalytic group. My next question is, what is the function of buttressing group and catalytic group? Answer. Buttressing group is meant for supporting the substrate, whereas the catalytic group is able to weaken the bonds of molecules by electrophilic and nucleophilic forces. Next question. Name a few factors which affect the activity of enzyme. Answer. A few factors which affect the activity of enzyme are temperature, pH, poisons, substrate concentration, product concentration, and inhibitors. Dear students, I am confident that you have learnt enough about enzymes and enjoyed the lesson as much as I did. Looking forward for the next class. Thanks for your attention and see you next time.